Within array, normalization techniques are important for those experiments in which we are using those differential dyes. So since we are using those different colors, and there is a tendency of different RNA to bind differently with either of these two dyes. So we need to do this normalization as far as these data is concerned. So biases are because of, as we said, two dye systems, CY3 and CY5 labels, they differentially incorporated at different abundances. CY3 and 5 have different emission or response to lasers at different abundances. So that is another factor that can disrupt your signal intensity or your count of those intensities and later on your results. CY3 and CY5 emissions differentially measured at different intensities, so it can also be an important aspect. The intensities can vary between regions, different regions, so arrays not horizontal when scanned. So when you scan them, they are not like in the way the experiment is performed. So different regions, they might affect or might reflect different intensities. Okay, so while you are selecting those genes in order to do those microarray experiments, the set of genes must be identified. You might have all genes on the array, so it assumes that small proportion of genes are expected to be differentially expressed. So you take all those genes, but within those genes, you have a smaller proportion that is the differentially expressed. And there is a symmetry in the expression of the up and down regulated genes. And remaining genes which are not differentially expressed, so they have a constant expression. So those constantly expressed are housekeeping genes. They are expected to have a constant expression across a variety of conditions. So we take all those genes, then we need to identify the genes which are housekeeping genes, so which are always there and whose expression is a constant expression. So we need to have them in the experiment. So housekeeping genes, they normally tend to be highly expressed, and they may not be the representatives of other genes of interest. So those housekeeping genes, there must be some technique in order to avoid them, or in order to remove them, since they are taking your bulk of your resources. They are highly expressed and most of your energies are going in detecting them, whereas they are of little use, since your interest may be in most of these experiments is what genes are differentially expressed. So for example, you give a treatment and then you compare with a control, but you find nothing in case of these housekeeping genes. So in generally like 10 to 20% genes are differentially expressed. So what about those remaining 80%? Obviously they are consuming your bulk of resources. We can have controls. For example, we can have spiked controls or titration series of control sequences. So what we do over here is we take synthetic or foreign organism DNA sequences and we put them into messenger RNAs at equal amount, and then we can have equal red and equal green intensity. So in this way you can control those biasness towards attaching with those differential dyes. In the second scenario, you can take the spots of the same sequence at different concentrations, and then your red and green intensities, they are expected to be equal across different concentrations. So you can have different concentrations of the sequences, and then you can see the red and the green, how they are behaving in those different intensities and on those different concentrations. So where you can try to neutralize the effect of those dye biasnesses. So all methods assume that most of the genes are not expressed. Otherwise, there is another thing what we can do over here is we can use a reference sample, and we can control, or we can have the spiked sequences as we have seen earlier. MA or RI plot is the scatter plot of log ratio against average of log intensities of each features. These are really important plots. Once you take those messenger RNAs, you put them on slides, and then you count their intensities, and after that you get those intensities and convert them into the log ratios, and then we take the average of the intensities for each feature and you plot them. So we call it as an MA or RI plot. So each point on graph, MA graph, is a feature. We have x coordinate, where on the x-axis we put the average intensity of CY3 and CY5, Y is the log of CY5 by CY3 ratio. So we take average intensities of both of them and plot them against the log of their fold change. We call this as fold change also. We'll talk about that when we talk these differential gene expression studies. How many folds, how many times the expression goes higher in CY5 versus CY3. So basically, geometrically, it's related to the log of CY5 against the log of CY3 in the scatter plots. So here is the MA plot. So we have M, which is log 2 of R minus log 2 of G. R is red and G is green. We can take A as log, both log 2 plus actually the average of both of them. So we take M on our y-axis, we take A on our x-axis, and in the middle we have a line that separates both of them. So you have a zero line. So in an ideal situation, we want that our data points should be close to this zero line. We perform regression of those log ratios on the average log intensities, and then for each feature, the normalized log ratio is the observed M or log ratio value minus the predicted M value. So here is those MA plots for 5 arrays. Before normalization, we see that patterns are kind of, most of them, they are away from that zero line. We normalize them. And here our MA plots are quite, fair, where we have the distributions centered around these zero lines. There is another normalization procedure called as lowest normalization, locally weighted scatter plot smoothing. We fit a smooth curve. We use robust local linear fit. So here, since I'm talking about linear regression or those kind of intensive statistical tests, so you fit up a line across these data points, and then we can try to, bring them close or minimum. We try to do some transformation so that they should come close to that best fit line. So we see in this section that biases and the experimental errors, they must be removed, and most of within array normalizations are for spotted 2-die experiments.